Okay, welcome back. We start again. And our next speaker is Dr. Michele Migliore from the National Research Council in Palermo. And I guess he's the best one to explain you how it's made, the single cell modeling. Michele,
thank you. Thank you, Paolo. Actually, I split my talk in 3 parts. One, which is this dedicated to single cell modeling, and there is already a YouTube version of it. So if you look for Michele Migliore Human Brain Project, YouTube, you will find the talk, this part of the talk. The next part is on how to use the knowledge that you will get from this first part to do some science, some additional science. And the last part is on circuits. How you want, what you have to do to implement, rather than single cell, a network. And in this particular case, a realistic network. And when I say realistic, I mean things that you can model, implement, and do the simulation, and then compare the results directly with experimental data, with experimental recordings. Okay? So, please interrupt me anytime you feel that you need more information, because of course I don't know your background. I assume that since you are in a basic neuron course, you don't know much about modeling, and you don't know much about coding, and all the problems that there are in implementing a computational model of a biological system. By the way, Paolo talk before was perfect and very nice, by all means, but especially because it allows me to introduce things that you already should have learned by the previous talk. So stop me so I can fix and try to figure out what is wrong, what I did, and so I can help you in understanding what I'm trying to convey. Because as soon as we go on with the talk, things are going to be more and more complicated. Okay? Okay, suppose that you have a set of experimental data. That's easy. I guess that you already have been exposed to the lab and do some kind of recordings. If not, this is what you can basically record from a cell. In this case is a CA3 pyramidal cell from the hippocampus, but it could be anything. The reason why we did this kind of implementation is that one experimenter came to us and said, okay, look, I'm recording from the same population of neurons in the hippocampus CA3 regions. They are recording from pyramidal cells from the CA3 region from the hippocampus. Rat or mouse, I don't remember. And they found that if they record from different cells, not only there is some variability in the recordings, which is normal, okay? But there are, and those are quantitative differences between one record and even if they record from the same neuron after 10 minutes. But they find also some qualitative changes in the firing rates, in the firing behavior of the neurons. So for example, in this case they found a few neurons firing weakly adapting, which means that the difference in time between successive spikes increase with time. Every time you have a spike, the difference in time with the previous is a little bit more. This is adaptation. It's called adaptation. Okay. In other case, under the same current injection. Okay. So we are talking here about the same current injection, but on different neurons. They find it a firing which was delayed with respect to the current injection, to the start of the current injection. In other case, the adaptation was much stronger. In other case, they find bursting. Okay? So these are the different firing rates that we want to model and explain to the experimentalist what is the basis for this change in the firing on the same neuron population. Okay? So, in order to do that, of course, you need to have the data, and we have the data. They are not enough, but I mean, just one example is okay, because we want to have a conceptual idea of how to change and what to change to have the different kind of patterns. And we need morphologies. And again, I know that yesterday you went to the Neuromorpho website where you can download the morphologies. In this case, the experimentalist gave us the morphology that he reconstructed on his lab. And this is one case of the pyramidal cells. So, luckily enough, I can skip all these slides because you already know everything about, no? Okay, so what we do if we want to model a single neuron. We take the morphology and then we split the membrane in pieces small enough that when you do the implementation with the equations, you can consider that small piece of membrane, let's say this, in this case, as isopotential. Who has done some calculus in his curriculum? Uh-oh. So if I say equation, you begin to shiver or what? I mean, you need to deal with equations, okay? So since the system is kind of complex, then you have a system or more or less complicated ordinary differential equations, and you integrate them numerically, okay? If the section that you are considering it is not small enough spatially, then you will end up, for example, with this dendrite, which is very long. It would be up to 100, 150 microns. And if you don't segment it in pieces small enough to be considered isopotential by the mechanism that integrates the equations, then you are going to introduce a lot of errors. Because, for example, if you inject a current here, Paolo has shown you before that the depolarization that you get is dependent on the distance. It decreases. But if you assume that it's an entire single piece, you are going to deal with it as it was only a single piece of membrane, which is completely wrong. Okay? So you need to split the dendrites in pieces small enough to be considered isopotential, so to reduce your errors. There are very easy ways to do this in NEURON, and probably you will do it during the course or not, I don't know. But in general, the best way to start with these things is to go to ModelDB, and I guess this afternoon you're going to talk about this. It is a site analogous to Neuromorpho, except that instead of having only morphologies, you have the entire model with everything. So you download the zip file, you compile everything, and you can run the model, you can make modifications, and then you can play with it from scratch, basically. Yes?
Are those numbers like the width and the length?
Yes, the diameter and length.
Okay.
Yeah. In this case, they are arbitrary. But they, basically, when you have the morphology, you will know everything. You will know at least the length of the entire piece of dendrite and the diameter. Because in the Neuromorpho, when you download the morphologies, the experimentalists will not tell you the different pieces. They will just give you information about the dendrite between two bifurcations. And it is up to you to tell neuron, look, this is too big, segment it in multiple pieces. Okay, I can skip this because you already know that the next step is to add passive properties in such a way that we can deal with the neuron, the propagation of signal, as an electrical circuit with basic properties of the membrane, like membrane resistance, capacitance, and things like that. Of course, it is not so simple, because on top of these passive properties, you need to add the active properties following this is an example of channels. The membrane, as Paolo told you before, is covered with ion channels. Okay, these channels open and close according to different functions of voltage and time, in such a way that you have the interaction between the sodium and the potassium channels, and they generate an action potential because the sodium is much faster than the potassium and things like that. But this channel are very small, but can be visualized. This is a tunnel microscopy. You see here two channels. One is closed in this case, and one is open. They actually have a shape of a hole in the membrane. Okay. I don't know. I don't go into the details of the Hodgkin-Huxley equations, but the important fact, the important information for you is that basically all ion channel kinetics, the opening and the close, activation and inactivation, are based on the Hodgkin-Huxley equation or some kind of formalism which is very similar. And what you get is the numbers that you see here, minus 25, 10, minus 1, 18, and things like that. Those are numbers that the experimentalist gives to you. So you go to the experimentalist will tell you, oh, I need to check that my ion channel, my potassium ion channels works in the way I think it works in my experiments. They will give you all these numbers in such a way that you can replicate the activation and inactivation properties, they call kinetics, of that specific channels, and you can include it into your simulation. It is relatively simple for beginners because a beginner just go to the ModelDB and download the file. It is called a mod file. Did you talk about mod files already? There are files in which there are the equations already there. So you take the mod file and you insert it into your compartment, and it is ready to go. You don't have to go into the small pieces and the details of the code. So we have here sodium, potassium, leakage in the case of Hodgkin-Huxley, but of course there are, I would say, at least 20, maybe 30 different types of channels, maybe even more, and they are slightly different from neuron to neuron, and from a neuron in a given region to another neuron in another region. Okay? And the way they work is basically determined by the genetic of the channel. So, for example, there is one channel which is called IH. It doesn't matter that you understand now what it does, but it is controlled by 4 genes, H, N, and 1, 2, 3, and 4. So each neuron is composed by a combination of these 4 proteins. Okay, and according to the neuron and where it is in the network, this combination changes, and if it changes, also the kinetics of the channel is going to change, and this has a lot of potentially a lot of consequences for how the neuron will fire. It depends on the ion channel. So, okay, that said, let's start from a CA1 pyramidal neuron. Why? Because the CA3 neuron that is our reference at this point has a problem. It is difficult to record from, especially if you want to do patch clamp. Don't tell me why. I never done an experiment in my life. Okay, but if an experimentalist tells me it is difficult to record the channels in the CA3, I trust him. Okay, which means that I need to figure out how to implement my channels. Okay, the closest thing to a CA3 channels is a CA1 channel, which is a population of neurons, which is the target of CA3 neurons in the hippocampus. You know the hippocampus morphology, shape, and things? I assume you do. So the CA1 pyramidal neuron, for which there are a lot of information, and there are plenty of models on ModelDB, plenty of papers describing the kinetics of the channel into CA1, and say, okay, let's start from the channels in CA1 and use those into a morphology of CA3 neuron and see what happens. Okay, so this is what we did. We can run the simulation. And by the way, these models are all in ModelDB, so you can figure out where they are and you can download and replay on your own. It is very small, but let's see. Don't be scared about all these panels. They're just because I want to show everything here in one slide. So basically, let's start from this. Okay? This is the basic experimental data. Can you see it from behind? This is the basic experimental data, the simplest, if you want, the simplest trace that you can model. It is a weakly adapting, and the action potential, standard action potential, we are not going to care about exactly the shape of the action potential. We just are interested on the spike times. Because when you go and would like to do the information processing, you are basically interested in the firing rates, spike times, and things like that. Are they going to study plasticity?
No, in this course.
Okay. So, but I need to talk on plasticity at some point.
Yes, the last paper will be on.
Okay. Anyway, so this is the basic line, the basic trace. And so we put channels, and when we say we put the channels, usually the only thing that we change in trying to figure out if we can model and implement the same trace is to change the peak conductance of the channel. Only in the extreme case in which we see that it is really bad, we begin to change the kinetics. Okay? This is because it has been shown, or it has been suggested by some experiments that basically ion channels are the same everywhere. They change a little bit the kinetics, but basically they're the same. So we change the conductance, we see if we can fit the trace with the conductance, and then if we can't, we begin to change the kinetics. Okay, so Ca1 channels, sodium, and two types of potassium channels. They have different kinetics. Let me show you here. Sodium channels, you saw them before with the Powell. This is basically Hodgkin type. This is a KDR, this is delayed rectifier of the squid axon, basically with the properties of the Ca1 neurons, which you have only activation and the time constant related to the activation. Of course, there was not enough time to go into the details of the kinetics, but when we show these kind of plots, again, those plots are implemented using experimental data, okay? And those plots are steady state curves. They are not dynamics, they are steady state. It means that, for example, if we start from -60, the resting potential, usually close to the resting potential, and we depolarize the cell very fast, let's say that with the current clamp or the voltage clamp, and you go to -30 or 0, it's better than 0. So you do a 60 millivolt change in the somatic potential. In principle, your channel was at 0 activation, so it was practically closed. It should go to a, let's say, 10%, 20% activation. It does not go to this state instantaneously. It takes some time, and the time is described by this curve here. Okay? So, sodium, potassium, delayed rectifier, A-type potassium channels, we just put some numbers here, and let's see what happens. This is another simulation that is running on my laptop. This is very easy because NEURON is very efficient. Okay, so you see this configuration in terms of current injection, duration, is similar to the experiment, but we are far away from the experiment. So we need to begin to change something in our peak conductance to try to fit those traces. And you see that there is already a problem here that cannot be solved with the conductances alone. And the problem is that the threshold for finding the actual potential here is that of the Ca1 pyramidal neuron. These type of neurons have a threshold which is around -30. Okay? There is no way with these kinetics to fit this threshold, because when you go from -60 to -30, your sodium channels is going to close, and it's closing very fast. Okay? So there is no way in which you can get a threshold to -30 by simply using this channel and modify the conductances. As a matter of fact, you can begin to say, okay, exchange this, exchange that, potassium, KDA, Ka. At the end, what you can do is reaching a situation like this. This is the trace, and this is the best we can do with the C. Yes?
No,
Oh, the best we can do with the Ca1. First, second, third, maybe fourth, but this is variability. So let's say that this is more or less okay. It is not okay, of course, with the threshold. So what we have to do to fit at least the threshold, because it is important, the threshold at which a neuron fires, it is important because when you begin to get synaptic activation on the neuron, and excitatory synaptic activation, the membrane begins to depolarize, And it is the depolarization which determines when the neuron is going to fire and then transmit information to the next stage into the network, okay? So the only way to do this is to assume that in these neurons, their channel kinetics are shifted, okay? And when I say are shifted, I mean that we take everything and we shift backward, in this case, on the positive side, in such a way that the threshold now can be up. So let's try this. Okay, let me erase this. This is again the thing. Now look here. This is the current situation for the channels, okay? And there is also this number here, KDR shift, Ka shift, sodium shift. This is what we can do if we shift by 24 millivolts the kinetics for the sodium KDR and Ka. Okay? So now we have a good agreement. We don't care about the perfect agreement, because if we get another trace, we are going to get a slightly different spike times. Okay? So we are okay with this. The other problem is that this is a weakly adapting. Weakly adapting means that you need to basically figure out a way in which the spikes are going to be delayed and delayed more and more as you go on with your current injection. It means that this timing is different from this and indifferent from that. Okay, it is going to increase for adaptation. Again, there is no way that you can get this kind of behavior using only these three channels, because none of these three channels has the right kinetic property to implement, to show the adaptation. Why? Because you see the difference between this spike and this spike. It is about, let's say, 80 millivolts. 80 milliseconds. Here is about 100 milliseconds. But if you look at the time constant of these channels, they are all between in the millisecond range. Maybe this is 30 milliseconds, but this is 12. So the time constant is too fast. By the time the neuron went back to the original resting state, the channels are back to the original conditions. So they cannot work for adaptation, because adaptation needs something that is still open when the neuron repolarizes. Okay? And of course, nature has thought, okay, I need to implement another channel, another type of channels. Okay? Channels which have a time constant long enough to take into account adaptation. So let me go to the next slide. Let me close this simulation here. Okay, don't be scared about all these channels, but they are in one way needed to fit this data. These are the original, not the original, these are the shifted version. You see that they are shifted by 24 millivolts to the right, sodium, KDR, and Ka. And now you see that there are two other channels, Km and Kca. Those are called, in particular, Kca. There are two additional potassium channels, one of which, the Kca, is dependent on calcium. Okay, so I don't want to confuse you more than already I did. So I'm not going to talk about the calcium-dependent potassium channels. I'm going to talk about this, the Kn, which is a very simple kinetics, only activation, but it has a 250 milliseconds of time constant at the physiological temperature. So what happens? It happens. Follow this line of reasoning. You are minus 60, or let's say minus 30. The current is slightly open. You go with an actual potential to plus 30, and you open completely. You tend to open the potassium, the Kn. However, according this time constant here, it takes several tenths of milliseconds at that potential, plus 30, for the potassium channels to open completely. But before it can do that, within 1 millisecond, the KDR shuts down the action potential, so you close and you go back to minus 30. Okay? So what happens is that at each action potential, there is a kind of hysteresis. The channel tries to open, but then it goes back, and when it goes back, it stays 200 milliseconds, it takes 250 milliseconds to go back to the original value. So it remains open a little bit with respect to the previous spike. And if you do this spike after spike, you are going to increase a little bit the opening of this channel, and you get the adaptation. Let me show you what happens. So it's kind of a short memory process. Weak adaptation with KM. Okay? So let's try and see what happens if I take out the KM. No. No, that's okay. Let's go do some. Let's do it from the beginning. Okay, so KM. As a matter of fact, I can raise everything here. Do the attention. Let's reduce this. Now I reduce the KM, so you have less adaptation. So you see that now that is missing the spike times because there is not enough potassium to keep the cell from firing. Let me stop this and go on the other side and increase a lot. Well, not a lot, but so I need to do this, and then that KM. Okay, instead of 58, let's say 9. So I've increased the conductance. The first spike is okay because the channels has not opened yet, but as soon as there is an action potential, it will open more than before. And so you are doing too much adaptation now. Okay? So there is the optimal amount of Km which is able to reproduce this data. The same with the calcium-dependent potassium channels. I'm not going to show you the results for this particular example, but we need to do it anyway for the bursting and the strong adaptation. Because when we go to strong adaptation, in this case, it is not possible anymore to take into account this strong adaptation with only the Km channels. Okay? Simply it cannot, because either you get too many spikes or too few spikes. So you need to introduce also another type of potassium channels which has a memory effect, short-term memory effect like the Km. In this case, it's the calcium-dependent potassium channels. So if I want to do only with Km, this is the best I can do. Okay? If I want to do it only with the potassium, for the calcium-dependent potassium channels, this is the best I can do. So the white and yellow, none of them is similar to what I have in the experiments. But if I mix them, a little bit of Km and a little bit of potassium-dependent, calcium-dependent potassium channels, then I can get this effect. Okay? Now we can go on and play with the numbers, and you will see that each change in the activity in the potassium in the conductances will give you different types of firing activities. Okay? So basically, KDR is able to act on the spike times. Increases the spike time, but it cannot give you adaptation. The Ka is a little bit delaying, but it has other important effects on the pyramidal cells, but we are not going to discuss it now here. The Km and the potassium and the calcium-dependent potassium channels are acting on the adaptation part. Okay? Let's go to bursting. The bursting is the most difficult to model because it is a kind of unstable condition. The neuron does a lot of firing and then stops. This is the burst. Okay, so for the bursting, we just need to, we found that it is similar to the adaptation, except that the contribution by the different channels is different. So we can be able to model the burst, okay, by changing these numbers. And of course, I'm not talking about the delayed version, but there is a specific current also that takes into account the delay of the first spike. Okay? So now you have seen here 3-4 different channels. With those 3-4 channels and calcium, you can model basically all the behavior of a neuron, of all neurons. Okay? But if you look and if you talk with an experimentalist, with a neurophysiologist, which does the patch clamp and explore the channels, they will tell you that there are hundreds of channels. Okay? So, and they will adjust the experiment or the experimental protocol or the bath or the temperature in such a way to amplify the effect of a specific channel that they want to study. This does not mean that that channel has in fact a significant effect on the firing rate or properties of your neuron. May do something else, activate second messengers, or do some kind of other things that affect the subthreshold integration and things like that. But of course, I will not go into those details, okay? Because otherwise you will begin to scream at me. I want to do something more high level in the next part. Okay, so the interesting point here is that once we have a model for all these traces, we want to know at the end what we know about the neuron, what we know about the configuration of the different channels that are needed to implement all these different properties. So what we did is to run a bunch of simulations. And basically several millions, 21 millions. We went, of course, to a supercomputer. We dumped all the parallel stuff that does the 20 million simulations in a few minutes. We got back the results. Basically, what we changed was the conductances of these channels, the input current, and we, for each case, we classify automatically the firing in terms of adapting, weak adapting, strong adapting, delayed, bursting, and things like that. And then analyze the results. So how many different combinations of channels will result in bursting? How many in weakly adapting? How many in strong adapting? And how they are correlated, okay? Okay. Let's try it. These are the different cases. You see the conductances that are changing, different combination, and on the left is the kind of bursting, the kind of firing that we have. Okay? So we analyze all these results, and we analyze the different combinations of the channels that we need to get the different. Of course, some configuration are ugly. Okay? let me see, are ugly just as you get in the experiments. So if you have done some experiments, don't tell me that you sometimes don't find ugly cells with ugly traces. This is exactly what we get in the simulation. Now they are all good, of course, but there must be some really okay, never mind. So basically, what we did is to analyze the results, and the results are this. Delayed. In order to have a delayed cells, of course, we need KD, which is a specific potassium channels with a kinetic adapted to have a delay at the beginning of the trace, at the beginning of the firing. Notice that it is impossible to get a delay without the KD. You can have a different combination, but there must be a KD. Okay, so this is a kind of a trivial case, but it is important, because if you have a recording in which you see some delay, then there must be a KD expressed in that neuron. Otherwise, you don't have a delay. Adapting. The adapting case, you see that in 95% of the cases, the calcium-dependent potassium channel was higher than the Km. This is again a prediction of the model. If a recording shows adaptation, either strong or weak, then the expression of the calcium-dependent potassium channels must be higher than Km. The opposite is for bursting. The KCA is lower than Km in 80% of the cases. Okay, so you now begin to see that each neuron can show different properties because the channels are configured in different ways. And this is important because you don't need to read all this stuff, but basically what I'm trying to say here is that there are going on in the neuron, in the molecular pathways that Paolo showed in that beautiful slide, the Roche molecular pathways, a lot of processes undergoing biologically in any cell, the neuron included. And this has been shown to change also the kinetics of the channel. So each neuron, based on its position in the network, it is going to express in a different ways the channels on its membrane to fulfill the specific role in the network in the specific place in that region. Okay? But not only that, since the system overall must be very robust, okay? Sometimes I do the parallel with the artificial intelligence. So if you do, maybe not artificial intelligence, but what is called neuromorphic hardware. You have chips that reproduce some kind of algorithm, okay? Well, if you go there and erase one transistor, you can expect what happens. Everything stops working. If you erase one neuron in your brain, who cares? Unless that neuron is specifically involved in some very special recognition problem, okay? So there must be some natural way in which a neuron can say, okay, I don't need to have exactly this configuration of channels in order to work in a specific way. There must be some possibility to have variability, as it has been shown experimentally. If you record from different neurons from the same population and you measure the channel conductances, you will see that there is some variability, okay? So we were interested in figuring out to what extent this variability is important or not. So what we did is to take a few morphologies, different morphologies from CA1 neurons, 34 morphologies from pyramidal cells, 34 morphologies in general, 19 from pyramidal cells and 15 from interneurons, okay? And for each of them, not for each of them in particular, but we got also from the experimentalists, not for those particular morphologies, but from other morphologies, about 1,500 experimental traces, 107 electrophysiological features were measured in each trace. What it means is that, let's look at for example, this red trace, okay? We have a way, I can show you in a few slides, a way to analyze these traces and extract the electrophysiological features, which means amplitude, spike times, shape of the action potential, resting potential, and some other things, okay? So for each trace of these 1500s, we extract the electrophysiological features. It is like what we did before for the single traces. Adaptation, bursting. In this case, we were much more specific in the kind of features that we want in our model, okay? We run it through an automatic program that does optimization following genetic algorithms, and it gave us, for each type of neuron, pyramidal cells adapting, interneuron adapting, interneuron bursting, interneuron non-adapting, and so on, to give us all the many sets of conductances which were consistent with this data. So in our many ways, I can model the same properties by changing the peak conductances. This is the key point. These are a few examples between experiment and models. So we got a good representation. We assign a score, and I mean, this is a kind of complicated process. I don't want to go in the details here, but we got this picture. So now, don't look at the details, but look at the colors, okay? So, in the x-axis, they are different optimizations, different ways in which we can reproduce the average firing properties of the pyramidal cells adapting, or the interneurons non-adapting, bursting, or adapting. Okay? So, for example, in this case, for the pyramidal cells, we have 160 ways in which we can rearrange the conductances of the channels that we are using, which are on the left side and the right side too, and getting the same firing properties that we observed experimentally for this type of neurons. Okay? And the colors means simply the normalized value of the conductance. What this graph tells you is that in this particular case, for example, there are channels, the IH, also some passive properties, which needs to be very high. Okay? Which means that if you lower the conductance of this channel, you are not going to get a good representation of your experimental data. It means that those channels must be high. Okay? Analogous here, the Km, they must be low. Okay? So, this and the same with other interneurons. What this means scientifically? Scientifically, it means that if there is some pathological conditions through which that specific channel is not expressed at the level in which it should be, or it must be, either high or low, you get a pathology, because the neuron does not work as it's supposed to work. Okay? And since we have a model, then we can think of doing a model of how to restore the original conditions by modulating the kinetics of the channel, because we know that there are pharmacological drugs or agents or compounds that can adjust the kinetic of the channels. So we can predict or suggest the drug company to how to modify the activation or the time constant of a specific channel in case it is mutated. For example, the Km is very involved in the epilepsy. Okay, it is known that there are papers that we publish with a group of doctors studying Km in babies. You know that babies, not always, but quite often, they have seizures when they are small, when they are young, and then this disappears with age. But then they in very rare case, they measure the Km channels and they found a mutation. in those babies, at least it was 1 or 2. But of course, I mean, human studies are very rare because it is difficult to go and do invasive studies of brain of human. But if you are volunteers, I'm sure that they will test you. Okay. So they found that the KM had a mutation with respect to babies not having seizures. Okay. So this showed that even in humans, the KM is important for epilepsy, and it was known from rats, but they showed also in humans. So this is the kind of studies that we can do with this. I'm showing you the paper that we published not because of the paper itself, but because it allows me, if the network works, to introduce you to the what we call brain simulation platform, or the Human Brain Project, in which you can do your own experiment without downloading anything. No neuron, no Python, no strange and esoteric things on your computer. Just go into the web page and run simulations. Okay. Of course, you need to have an account. It is a site in which you have an account. So, for example, this we put here everything, and so the morphologies. It is going to ask me for an account. I have an account, and but before you go that, I want to show you how you get an account. Okay, let me see. No, okay, so this is the morphology for this particular case of this particular simulation. If you go this is a website, so it is open, it's free. To view the morphologies is easy. You just click and look at the morphology. The blue is the axon, the red is are the dendrites. However, if you want to run the simulation, these are the electrophysiological traces. Everything is here. This is the model link test simulation. So, for example, I want to do this. Now I'm calling the server that is on the back end of the Human Brain Project. Yeah, okay. So I'm stopped here just because I want to suggest you to send an email here, support@humanbrainproject.eu, asking for an account. It is free. Okay. And this will give you access to the platform where you can run your own experiment, build your own models, and do a lot of things that I think it is very useful. And if you have some feedback, we appreciate if you can send us bugs, things that you want to improve, and things like that, because we want to extend and disseminate this platform as much as possible. So I'm going to put my account here. Okay, now I'm in this particular section that is going to run the simulation on the server of this particular neuron which I clicked before. And this is all online. It is doing something, so I better not interfere with it. While things, I can go to the simulation platform to show you what it does. As we hear, okay. So this is the neuron. We are not showing the axon because it is not important. We want to do the dendrite. We want to do the in silico experiment on the simulation that we did for the paper, for example. So we can actually switch between the morphology or the what is called the dendrograms to show you the morphology in terms of how many dendrites there are, which length, and things like that. And so if you do the simulation here, you just click, and let me see. No. And it does the simulation, shows you the results, you can download the traces and things like that, okay? Without having neuron installed. This is done on the server, and this is a web browser. But the brain simulation platform does much more than that. Once you have the account, you enter here, you go to there are several platforms, neuroinformatics, brain simulation, high performance, medical, neuromorphic, and neurorobotics. And we are in particular involved in the brain simulation platform. And you see that there are several use cases, online use cases, molecular level, cellular level, trace analysis, morphology analysis, and so on. These are all free. You can run them without problems. You can run single cell simulation, optimization. There is also a free account to give you computing time on supercomputers. Okay? This could be done either through the Neuroscience Gateway. I don't know if you're going to introduce the Neuroscience Gateway to them. No?
So far I didn't think about that.
Because the Neuroscience Gateway will give you free computing time. Okay? But we also have a service account, which means that we can give access and computing time to people that are interested in exploiting the platform or they want to collaborate with us, except for the large area like the hippocampus, cerebellum, basal ganglia, in which it is easy to consume 1 million hours without you knowing how. So then we need to be very careful. But anyway, I think that it would be very useful for you. Okay, so this concludes. These are just slides in case the network did not work. Let's go one step up. Now we are tiring of playing with single cells. We want to model our own brain region. Okay? So of course you can understand that this is a difficult situation, especially if we want to do it very closely to the experimental data, because we need a lot of data. So in principle, what a physicist does, or a theoretician does, is okay, I don't care. I think of my brain region as a black box. I give an input, I get an output. If I have enough input and outputs from the experiments, I am smart enough to implement a deep learning, an artificial neural network function. I find a function that will generalize good enough in such a way that for any input it will give me the right output. It works, because if you look on the internet, you will find robots which looks amazing. Okay? The Google car, or the dogs from Boston Dynamics, I don't know if you saw that looks amazing. But try to change the activity of the dog. Or try to put your Google car from the streets in California to your backyard in your home. The very first thing that, the two things could happen. Either it stops, it doesn't know what to do because it doesn't see it much, or it begins to bump against the wall to learn. Okay? Because it needs to learn first. But if you go from, I never been here. Yesterday was my first time, but I knew today where to go to the entrance. You can say, yes, you have 60 years of experience with your brain, and things like that. But it doesn't work this way. It is our brain, our hippocampus in particular, can generalize based on our past memories in a very flexible way. Okay? So what you want to do is open the box, look inside, study the internal wiring and working of each node, and then put back together, and then we can say, now we have the real algorithm. Now we can reduce the algorithm, put in a chip in the car, and then the car will behave like a human, more or less. Okay? So, from where we should start? We identify, of course, the region. There are many regions in the brain, but in order to be able to figure out something, you need to have a relatively simple sequence organization, a small number of cell types. They must be studied experimentally because we need the experimental data. It must be easy to carry experiment and compare results across species and humans, because you cannot do experiments in humans unless there are some specific particular cases, but you can do experiments on animals. And it must be involved in other brain functions, because otherwise it is not fun. And we found that the olfactory bulb, which in the humans is very small, But in the rodents, it is 20% of the brain, because of course the rodents rely completely on the olfactory sensory systems, okay? So we decided the olfactory bulb was our system. Okay, so do you know how it works, olfaction, in principle? So I tell you one thing. It is why it is important. So I'm sure that in your everyday activity experience, you have been under a condition in which someone is talking to you, but you are too concentrated to read or listen to music, or even without headset, right? You just ignore, because your attention is focused there. Well, this cannot happen with odors. Okay? There is no way in which you can avoid smelling an odor. And this is because the inputs from the olfactory bulb goes directly to the hippocampus, skipping the thalamus. The thalamus is the multiplexing region in which decide to which input you need to take to give priority. You cannot do this with olfaction. And this is also why the olfactory, the smell, is such a powerful evocative properties. I mean, when you smell something, everything comes to your mind. Images, voice, sounds, everything. Okay, this is because the olfaction must have priority over, this of course is a legacy from ours being a rodent some millions of years ago, but it's still valid for the humans. So there are some cells, the olfactory sensory neurons, which transform the odors into this electrical signal. The axon goes into the bulb, and then in the bulb there is a very simple organization in which I don't know if you can recognize, but this is the side view of a rat. Okay, this is the head. This is the mouth with the nose, and this is the olfactory bulb, and then there is the rest of the brain. So in one synapse, there is the odor activates here the dendrites of these cells, which are called mitral cells, and which are modulated by large population of interneurons, but then they send to the cortex the signal, which is clean, organized, and set up in such a way that the cortex in 100 milliseconds can decide to stay and enjoy or run for your life. Okay, so it is a very efficient system because it must be very efficient in recognize a danger or not. And this is the reason why we still use dogs in the airport. They don't use the machine. There must be a reason. And the reason is that all those electronic nodes that are around, they have a lot of false positives. They signal something which is not there because the sensitivity of the sensors that they use depends on temperature, humidity, with time, and it has not the flexibility. They cannot smell different things. A dog, you train a dog, and it's very effective. Okay. Okay, so these are mature cells. Of course, we need experiments. So we begin to collect a few mitral cells. And the basic circuit of the olfactory bulb is very peculiar, but it is in one way or another reproduced in other brain regions. So there is the so-called dendrodendritic interaction. So far, you may think that synapses are from an axon to a dendrite. In the olfactory bulb, there are basically very little axons, the one that the mitral cells that go to the cortex. Everything is controlled by synapses between dendrites. The dendrites of the so-called granule cells, which are inhibitory, and the spines that are on the mitral cell dendrite. And it works this way. The odor generates actual potential on the mitral cells, on the distal dendrite of the mitral cells. It propagates to the soma, and then it propagates to the lateral dendrites. The mitral cells has a very peculiar 3D shape because the dendrites must follow the bulb, so they are kind of ellipsoid shape. But then they need to talk with the olfactory sensory neuron. So there is an apical dendrite which comes out of the soma and goes up to the surface of the bulb to make synaptic contacts with the axons of the olfactory sensory neurons. But as the action potential propagates along the lateral dendrites, it spits glutamate, which is excitatory. And this excites the granule cell spines, which in turn, just for revenge, will spits GABA, which is inhibitory. Okay? So you have this kind of excitatory inhibitory activity, which is within a few milliseconds. And this occurs all over the place during the lateral dendrites. What are the consequences? Well, okay, so this is a famous set of experiments done in vivo on a rat. And this group, the Japanese group led by Kensaku Mori, did an experiment in which they were recording from a single neuron, single metal cell in a rat, during odor presentation. And they select the odors in such a way that chemically they are very similar. Okay? And in spite of being very similar, they generate in these mitral cells completely different responses. In this case, the mitral cells was recognized the odor was generating a lot of spikes. In these two cases, it was greatly inhibited. Okay? And so they suggest that the organization was something like this. Mitral cells, granule cells, excitatory cells, inhibitory cells, reciprocally connected. So what happens is that if you activate a lot this mitral cell and a little bit less these mitral cells, then you have a lot of activity in this and less activity here. But a lot of activity here generates a lot of inhibition on the other cell. This is called lateral inhibition. I'm sure you know it, right? Who doesn't know lateral inhibition? Don't be afraid. You all know lateral inhibition. Good. So I can go on. So the lateral inhibition generates, basically it generates contrast enhancement. Because if you have, if you have done some kind of artificial neural network, you know the concept of the winner takes all. In this case, the mitral cells, which is mostly activated, is going to send a lot of inhibition to all the other cells to which it has connected. And if they don't have the same input, they will be shut down because of the imbalance between the inhibition generated by these highly activated mitral cells and all the others. Okay? But there is another important, let's say, cognitive function for having this kind of circuit of excitatory and inhibitory cells reciprocally connected. Let me show you a simulation. Okay, here I have two neurons which have the same input. They are reciprocally connected, and now the inhibition is 0, so I set up the weight of the synapses to 0. And they receive the same input, so they are going to fire, but the input started at different times, so they are out of phase with each other. Okay, you see that they fire on their own. But if I add inhibition, they synchronize. Okay, now if I remove the inhibition, they stay synchronized. So it is thought that this is the basic mechanism through which you can recognize something, because the network is wired in such a way that if some group of neurons are reciprocally connected, then when they receive the same input, they are going to synchronize, and so you have a powerful output that you can make it to converge to some recognition cells and say, okay, here it is. Okay? And if you group together different ensembles of neurons, you can recognize different things. Even if they are overlapping cells, you still have this way a mechanism to recognize one object or another object. Okay, so let's try to model this system. Again, this is the basic configuration of the Kensaku Mori experiment. We have 3 mitral cells, 3 granule cells, and we set up different odors, odor 3, 4, 5, 6, 7, 9, until 11, And what this bar graph means is that, for example, for odor number 5, these cells get activated a lot. This is activated very little and very little. Okay, so odor 5 is the preferred odor for M1, odor 7 is the preferred odor for M2, and odor 9 is the preferred odor for odor 9. Okay? Now, if we do the simulation and present different odors, and we record from mitral cell 2, this is what happens. For odor 5, mitral cell 2 is inhibited because the preferred odor is for mitral cell 1. For odor 9, it is mitral cell 3 which is preferred, and so this gets inhibited. But for odor 7, it gets excited because it is his preferred odor. Okay? So we can reproduce the experimental data. In principle, we can be happy. In fact, we publish it. Okay? Except that the model is wrong. Okay? It is conceptually wrong. Why? Because if you look at the network here, I mean, it is wrong in the nice sense because we publish the paper anyway. So it is an important contribution to science, except that it was done by using a very popular organization of networks in the artificial networks world, which is we made an all-to-all connection. Okay? Each neuron is connected to every other neuron. This cannot happen in the brain. This is wrong by default, because you cannot have any neuron in your brain connected directly to all the other neurons. Your brain would be the size of the Empire State Building, probably. So there must be some kind of shaping the connections and decided why one synapse gets activated and another is removed from the network. Okay? So we went back to the experimentalists and asked them, okay, we need to know how the olfactory bulb is connected, because otherwise we cannot advance in our will to build the olfactory bulb. And so they did the experiments. They injected a pseudovirus with a genetic modified protein, and basically what happened is that they were able to visualize the synapses connectivity between mitral cells and granule cells inside the olfactory bulb. Okay? So basically they inject the virus here, the virus crosses the synapses, active synapses, and then is brought to the nucleus of the cells to which they are connected, and then it reproduces, so that cell become green. Okay? This is what they tell me. Don't ask me questions on how they did the experiments, okay? But they did experiments, and this is the result. So these are slices of bulb, and you see, I mean, the organization is, here are dendrites of the mitral cells, these are the soma of the mitral cells, this is full of granule cells, okay? There are about 40 times more granule cells which are inhibitory with respect to the excitatory mitral cells. What these pictures means, and I show you some more detailed view. Here is full with granule cells, okay? But only if a selected group of granule cells are making synaptic connection with these mitral cells, or with any dendrite which happen to cross here, okay? So basically what these results are telling us is that the connectivity inside the human, the olfactory bulb is not random, is not distance dependent, is not some fancy rules that are used in artificial neural networks. They are specifically connecting mitral cells with a selected group of granule cells for a reason, okay? And so what means is that the connectivity is so-called sparse and distributed. Okay, so basically this is a picture of the synaptic connectivity at that age of that particular animal, in that particular condition with his previous experience of odor presentation. If I take another animal, I don't get the same photo, I get a completely different configuration, but it will follow the same rule. There are so-called columns or preferred connectivity between granule and mitral cells. So of course, our next step as a modeler was, we need to find out what's going on, how we can get to this condition. So we set up a model, another one, another publication. So what we did here is, okay, we have a mitral cell with a long dendrite, which is, by the way, even shorter than the real mitral cells, because mitral cells have lateral dendrites which can be up to 1.5 millimeters, spanning the entire bulb. Okay? In this case, we set up 500 microns, and we set up also 50 granule cells below each segment of this dendrite with individual synapses, excitatory and inhibitory. And for each synapse, we set up a plasticity rule. I don't know if you are going to get to the last day with the plasticity, but you know that in order to learn something, your synapses needs to change. The weights, synaptic weights needs to change. So we assume a very simple synaptic plasticity rule in which for low frequency of activation, nothing happens. For intermediate frequency of activation, let's say for a few hertz and 30 hertz, the synapses depress a little bit. For higher activation, it potentiates a little bit, according to a sigmoidal function. So basically, it means that if you stimulate a single synapse with the right frequency for enough time, in this case 50 spikes, the synapse will go either potentiated to the maximum or depressed to the maximum. Let's look at what happens. Okay. So now you are going to see an universal explanation here. I can minimize this. So here you see the somatic activity of the mitral cells. This is the dendritic activation of the dendrite membrane potential at 350 microns from the soma, which is around here. Okay? Here you see the activity of the granule cells at 350 microns from the soma. And here you see the activation of the granule cell below the soma. But remember, there are 50 granule cells along the dendrite, and you are going to see their weights, excitatory and inhibitory weights, here. So, let's go. Where is the panel? I lost it. It must be somewhere. Here it is. Okay, let's go. Play, pause. I don't want to show you directly the results. Here we have all the synapses start at 0, of course, because we want to know how they evolve. So frequency of activation, there is nothing that is basically happening except that as the action potential propagates along the lateral dendrites, all the excitatory synapses are going to be potentiated because they receive a high frequency of activation, okay? But as they begin to raise, to be potentiated, they begin to activate the granule cells. You see here? Now they are strong enough, they are basically their maximum value, they are going to begin to activate supra-threshold the granule cells. So the granule cells are going to reply to each activation with an inhibition. And so their weight is going to increase too. Okay, so their inhibitory weight is beginning to increase. Be careful on what is going on at the soma and at the dendrite. In the soma, where the action potential is coming from the distal dendrites, the inhibition cannot do anything. So the soma is going to fire still at a very high frequency. However, as the action potential propagates along the dendrites, all the inhibition is going to affect the propagation until you begin to see some failure on the action potential. But when you see the failure, it means that the frequency that locally the synapses is affecting, is seeing, is within the depressing frequency. So what you see is this. The column. There is no magic here. It is just network evolution. I will show you from the beginning without interruption. You see here high frequency, low frequency because of the effect of the inhibition. excitation, inhibition, and the column like in the experiments. Okay? So basically what we are showing here is that those columns that you see in the experiments are a simple expression of the fact that the mitral cells belonging to that particular group called glomerulus in the past experience of that rat were highly activated, and then all the synapses were potentiated right there, not outside. Okay? Okay, so we have a model, we can build a model, we have a model for the synaptic evolution. This is a very simplified model. We want to do a 3D model of the pulp. Okay, so it is not enough to stop here. We went on and we asked Kensaku Mori lab to give us as many as possible experimental data on activation of the mitral cells, or the group of mitral cells called glomeruli, from the dorsal part of the bulb of the rat. The dorsal part is the most studied because they just opened the school and measure the things, okay? And they gave us, this is a bidimensional representation of the olfactory of the glomeruli on the surface. Each color means a different zones, but they gave us, don't look at the numbers, it doesn't matter, look at the map. These are the activation coded into 4 levels for 73 glomeruli, so for 73 locations in the dorsal bulb for 72 odors. Okay, these are complete chemical series. Okay, so these are experimental data. They tell us that, for example, this block, yellow block of glomeruli of dots here is activated preferentially for these odors. And there are other things. So basically the odors are mapped. These are monomolecular odors. So we implement a larger network in which we still have a very simplified neuron with 2 dendrites. Okay, we mitral cell with 2 dendrites. We basically made a topological organization of the system which is bidimensional by saying, okay, even if the mitral cells is here, we assume it looks like as it is here, we project all the connection on the 2 single dendrites, and let's see what happens. Okay, so even if you are going to see a 3-dimensional representation, it is still bidimensional because the mitral cells have only 2 dendrites. Okay, and this is the simulation of odor 39, which is the one with this arrow here. It is going to activate this portion of the bulb. There must be sound too connected to the granule cell firing. I don't know if you're going to hear. Let's see what happens. So this is, I think it is 500 mitral cells and 10,000 granule cells. The granule cells, of course, are the green one, and the mitral cells, I'm showing only the soma, are the red cones. Now it comes the odor that is going to activate here, and the network is beginning to learn. You see the noise? As it learns, synapses are going to grow, granule cells are going to emit spikes, and the system is going to self-organize to be able to recognize that odor. If we now learn another odor, the system will follow the input and will self-organize in a different ways, in such a way that you see this noise, the noise is going to be different for each odor. Okay? But still, this is not the real 3D stuff that we want to do. Okay? However, with this model, we were able to answer a lot of puzzling experimental questions that were at that point still dangling in the field. So this model was useful, but now we want to go to 3D. And so we asked a group in Switzerland to give us 128 locations for natural odors. And the natural odors are much more complicated with respect to the single odor, monomolecular odor, because they activate the bulb in a much more larger way. Okay? Anyway, so they gave us these 128 locations, And at that point, of course, we had the problem of having the real matter cells. Okay? And from the same, luckily enough, the Kensaku Mori lab, there was a guy, Kei Igarashi, that was able to give us about 10 reconstructions of mitral cells. It is a difficult job because each mitral cell takes about 1 month of work to be reconstructed because they have to go through image stacks. You know what it means? It means that since, in contrast with the CA1 region, pyramidal region, Purkinje, cortex, they are not flat. The mitral cells are real 3D objects, so they cannot do a slice because they are going to cut a lot of dendrites. They need to do the injection of the dye and then do the image, do the slicing, put everything together in register and manually adapt everything and get 3D reconstruction. Of course, we want to do thousands of them, so we cannot ask them to do 1,000 reconstructions, so we need to use those 10 cells to analyze their statistical properties in terms of average bifurcation probability, growing direction, and dendrite lengths and things like that, and build a software, an algorithm, which was basically synthesizing new morphologies, which were, however, statistically independent from the original morphologies. Okay, so at that point, with that algorithm, we were able to generate any number of mitral cells, which basically are indistinguishable from the real cells that they have in the bulb. So let me show you a few examples. So you see, each one is different from the others, but they are exactly the same statistical properties of the real reconstruction. And of course, then once we have this, we can build the model and do the simulation because we have also the channel kinetics of the mitral cells, so we can build the model. And now we are ready for the big model. Okay, big model, first of all, does not mean the entire stuff, but we started with the glomerulus, which is a group of 5 mitral cells in our model that receives a localized input, and but with interacting with a bunch of granule cells, following the experimental constraint in terms of synaptic probability, of the connection probability, and things like that. Okay, so this is a glomerulus. Okay, this is the cloud of associated granule cells. Now I'm going to show only one mitral cell to show the activity. And okay, this is a sniff every half second. You see that at the beginning, oh, by the way, let me explain you what is going on here. Of course, this is very simple. There are activation of the tuft, the action potential generated that then propagate along the lateral dendrites. Here you see a visually color-coded image of the weights of the granule cells. Okay? So with activity, the granule cells are beginning to spike, the weights are going to change, but you see that at certain point, as the weights increase, the action potential does not go through anymore. Let me see if we can spot one of these. Okay. It's not, yeah, here, you see? So the weights are growing and they are basically saying, okay, stop propagating the action potential because they use energy, we don't need them, we want to stay here because the connectivity must be local, okay? So after a few sniffs, the weights are doing the column and they're doing their job. At the end, we get, this is the experiment and this is the model, okay? So we found it pretty good as a reproduction of what's going on. And we did not impose anything. We just used the experimental constraint. We call it data-driven modeling, okay? We found also that this same organization does not depend on the specific plasticity rule that we were using. We can use spike-time dependent plasticity, Hebbian learning, or whatever, but the basic concepts still remain here. And the problem is that the question is that no matter how you implement the plasticity rule for your network, the important mechanism is the propagation of the action potential and the activation of the granule cells during the activity, during the learning procedure. It doesn't matter what learning you have, but as far as the granule cells learn something, they are going to hinder the propagation of the action potential, generating this kind of columns. Okay, the big system. Now we are ready to build our little box. Okay? So of course you cannot run this on a laptop. You need a supercomputer. In this case, we have the dorsal part of the bulb modeled with about 600 mitral cells, 70,000 granule cells, about less than 1 million synapses, about 1/28 of the real system area. And this is a system of about 32 million nonlinear differential equations. Okay, it is not that big, but it needs to be run on a supercomputer. For this particular model, 40-second simulation on 2000 processor, it requires about 9 hours and produces 10 gigabytes. For those of you who are doing parallel processing, this is a 99% efficiency, so it is very good in running and using all the computer time that we have. And this is the system. So this is only a part of the system because we have only one mitral cell for glomerulus. In fact, in the model there are 5. But and this is a typical simulation in which we activate these 3 glomeruli with a given order. At this point, you can figure out that we are able to run what we call in silico experiments. It means that we can do whatever the experimentalist can do, and even better, because we have control over the entire system. Okay, we can look outside, inside, select a few cells. We can do anything. This is a perfect experimental setup if you want. Of course, we are missing something because no model are perfect, like any experiment is perfect. But still, it is sufficient for us to see what's going on. Let me go a little bit further because I want to show you, this is kind of Hollywood stuff. I skip it, but I'm going to increase 10 times the speed of the movie, and this is going to be still 10 times slower than the real system. So this will give you an idea of how fast we process information, not only in the olfactory bulb, but in our brain, because this is, after all, a system that is similar to any other brain system. Okay. Okay, so this is 10 times slower than real. Okay? Okay, so now, what we can do with this system? One thing that we can do is begin to see how the network is self-organized. One of the very first thing that we figure out is that the synaptic connectivity of a bulb depends on the sequence of odors that we are exposed to. This is, seems to be trivial, but it is important to show and to understand why our olfactory bulbs could be completely different from each other, because it depends on our past history. So these are some examples. There are also cooperative interaction, competitive interaction, but it doesn't matter. But the real point here is that now we have a box fully open for us, and we can figure out what is going on. Yes?
Just a stupid question. Is the system naive at birth?
What? Say it again?
Is the system naive at birth?
We don't know, but it must be naive, except that when you form a synapse, the synapse is not going to have 0 weight.
Right.
It is formed with some basic weight.
So you think it's random?
Random, but small. Okay? And we tested also this with the model. It is the same. It does give the same results. Yeah.
Can I ask a related question? I'm only partially understanding what you're discussing, but you're talking about activity-driven plasticity and shaping of the pathways.
Yeah.
I'm wondering how that may be translated into morphology. So when you, for example, you are modeling, you built this model based on the morphology you were getting of mitral cells.
Mm-hmm.
Were those mitral cells naive? I mean, or in other words, were they from what age animal?
Oh, yeah.
In other words, is the architecture of the projections in fact experience driven? Or in other words, or another way of even putting it, can the connectivity kind of learning be then, shall we say, hardwired later into pruning, right?
Yes. So the answer is that yes, everything is changing with the life of the animal. In fact, the granule cells of the olfactory bulb is the basically only region in which you have a constant rebirth or changing configuration, because granule cells are taking out of the network and new are born and inserted into the network. So the connectivity changes constantly. We don't know why and what is the specific computational properties, but this will not change the basic functioning of the network. It may be change something in the information processing at any given instant of the life, but so for example, if you have mitral cells which are not yet developed completely, the lateral dendrites may be shorter than the final adult stage, and I guess that the age of the animal from which we got the morphologies were adult animal, and so everything could change, but the basic mechanism of forming the colons will remain the same. Okay, I don't know if this answers your question.
It's part of it, yeah.
So at this point, we have the full model, we have the full box. We can think of generating our function. We can call it an order operator. Okay? We have 700 mitral cells, 70,000 granule cells. We can build an olfactory operator because we know the connectivity between each cell with any other cells. Basically, we have a matrix with the connectivity and the weights. Okay? So it is just a matter of matrix computation, and we can build an order operator. Okay? An order operator is something that will give an output in the presence of a given input. It's that simple, except that instead of inventing the function on our own, now we have the model and we can actually write the right function. Okay? It is just a matter of figuring out the weights, excitatory, inhibitory, how they are connected, and we get the operator. So, at this point, the operator, I mean, you can go down with the operation, but it's just matrix multiplication. And at this point, for example, let's look at the operator for after the bulb learn banana input. Of course, as with your learning, this map is going to change and will give you what that bulb is going to be able to do in the presence of a given order. Okay? And the response is going to be, could be quite different from one order to the other order. And so we thought that we can use this as to build the prosthesis for the olfactory bulb. So basically what we did, we proposed, these have not been funded yet. We made a proposal. So we use supercomputer to build the full model. From the full model, we do a reduced model and we do the neuromorphic hardware, and then we can control an animal in vivo by basically cutting what is called the olfactory lateral tract, which is the bundle of axons that connects the olfactory bulb with the cortex. And instead of having the input from his own olfactory bulb, we can give it the input of our bulb model, okay, and see how this interacts in a virtual environment. And we can do this because, of course, with the full model, we cannot interact with the environment in real time because it is too slow. But with the order operator, we can build a chip. the chip is fast enough to interact with the overall environment. Okay, so let's go to another system very quickly, using basically the same line of reasoning. We are not as advanced as the bulb for the hippocampus, but still we are trying. So in this case, the hippocampus of the rat has a few million neurons. In particular, we are starting with the CA1 region, the full CA1 region. In the humans, there are about 20 millions of neurons, but with the rat, a few millions. Actually, in the CA1 of the rat, it has about 500,000 neurons. So it could be done. The hippocampus is important because it is involved in a lot of higher brain functions like learning, memory, spatial navigation. It is involved in a lot of directly involved in a lot of important diseases like Alzheimer, temporal lobe, epilepsy, cognitive aging, and you name it, the hippocampus is there. Okay, so the organization is relatively simple. It's not simple, but there are three populations: CA1, CA3, and DG. They are interacting, receiving input from the entorhinal cortex and things like that. I don't want to go into the details of this, but the symmetry is not in columns like the cortex, but it is like in slice. That's why they do the slice in the lab. What we are really interested in doing is to generate, it doesn't matter what these blocks are, but you need to know just what they mean. And they mean that we can build what we call a workflow, which is a series of steps which are the same independently of the region that you want to model. The details, of course, depends on the region, but in general, you have morphologies. You have you need to curate the morphology. You need to have the single cell recordings. You need to do a model of single cells. You need to get experimental data for the synaptic transmission pathways, synaptic types, and all of this are these blocks. Finally, you need to put the system together. We do it with a touch detection system, software. And then finally you get to the circuit simulation in which you can get the circuit, okay? So this is the circuit so far. What we really want to do is first implement 1/2 of the hippocampus and then reduce it into a chip that we can put into a robot, okay? So far the system has about 500,000 neurons, so it is much bigger with respect to the olfactory bulb. It is about 90 millions of membrane segments, and this means about 2 billions of ordinary differential equations, not counting all the plasticity stuff, equations and things like that. So one simulation time takes between 3 and 9 hours on a KNL system, supercomputer system, using 13,000 processors, 13,000 cores. And it produces 4 terabytes of output, okay? But again, and this is one simulation, as for the olfactory bulb, we can basically do whatever we, in silico experiment we want. Local field potentials, we can do imaging, compare the images with the EEG recordings. We can do single cell, multiple cell recordings, slice, like here, for example. And basically we have full control of the experiment. And we think that this is the way we should do, and we should go to make real advancement in, for example, treat pathologies and diseases, because again, here we have all the channels, all the dendrites, all the interaction, the synaptic integration properties and things like that. So we can connect directly with the laboratory and show what they can do to deal with this system. Okay, I think that I'll stop here. Let's see. No, okay, yeah, this is the reduction process. And I just want to mention that the hippocampus is a large project involving several different groups within the Human Brain Project, and we are just part of the group. And this is part of my lab. And thank you very much. If there are other questions, I will be happy to answer.
just a wild question on the hippocampus.
Yeah.
The hippocampus work that you're doing. I'm not even sure how to frame this, but just a rough idea, and that is one of the functions of the hippocampus is to describe in terms of spatial memory. The spatial memory is the idea of a place cell, right? And ultimately, to the extent that that is, I'm not sure. I don't follow the field, but I've heard there's some debate about whether that is whether the initial interpretation of place cells is really what people thought they were. But let's assume it was. Is your modeling, can it support the notion that from very, very complex inputs, right, what
maze
ultimately lead in the modeling to a specific local activation of the cell?
Well, this is what we are trying to implement right now. The first release of the model does not take into account external inputs because we want to know the intrinsic activity of the hippocampus C1 region. And this means being able to show that our model can reproduce basic facts, experimental facts on CA1 activity, which is traveling waves at theta rhythm and how they interact with gamma rhythm, and this is the basic. The very first thing is going to be adding plasticity and see under what conditions the place cells could be formed, with or without grid cells input, because you know that grid cells in the entorhinal cortex are driving some types. There are different types of place cells. The one that depends on the grid cells, which are basically coding for the head orientation in the world, and place cells that don't depend on the grid cells but depend on what is your memory, basically. Okay, and so we are going to test under which condition you get place cells formation, but we need to add plasticity, which means that instead of 4 gigabytes of data, when you want to follow billions of synapses, it is going to be complicated. So we need to we are still in the process of finding the right strategy to follow synaptic self-organization, network self-organization in the hippocampus. But we are working at the small circuit at single cell level to see place cells formation and how this could lead to spatial navigation. Because for spatial navigation, you don't need only single scattered place cells here and there, but you need to build sequences. Because the rat needs to know, now I'm here, I need to go there instead of there, and then when I'm there, I need to go this way. So this means a sequence of activation that needs to be reproduced by our model to say that this is a realistic representation. We are not there yet.
Okay.
Other questions?
Any other questions? Maybe you can address more questions to Michele during lunch.
Yes, in priority, because
So for an incoming scientist into your group, how long on average does it take to sort of get trained in the trade?
well, it depends on your background.
Let's say you have very minimal background.
So I tell you, this is part of my group. She is a chemist.
Mhm.
She is a theoretical physicist. She is a computer scientist. She is a biomedical engineer. She is a mathematician, physicist, mathematician, and she does the administration for the Human Brain Project, which is I thank forever. Okay, so it is very. It is not the learning curve that matter, but the motivation. Okay, as I tell every day to all of them, and they agree. If I don't see the blood in their eyes, it means that they are not working enough. Okay.
Okay, and at that level, how long does it take them to sort of be seriously involved in their part of the project?
Yeah, you can be involved in the project in a matter of weeks for the low level, for example, single cell optimizations. The first thing that we do. Single cell is the easiest one because you don't even need a background in neurobiology. You can do it while you work. But if you want to do, for example, the same if you want to do information processing, because you have the model there, you have a simulation, you don't care how we built the model. You just run a simulation and analyze input output. You have the spike, the input activity, you have the output activity, you can model anything. So to advance the field of the model, for example, to add plasticity, that's another story, because you need to be able to program in Python, be able to interact with a supercomputer, the things that get much more complicated. But to be productive in selected fields does not take much, unless you are a biologist. Okay? Biologists and the psychologists, I don't know if they are psychologists, they are simply not their field. Okay, so I was telling a story yesterday at dinner. I teach 2 courses, one in Palermo for computer scientists. They can do a simulation in a matter of minutes, better than I do. But when I talk about ion channels, they look like completely lost. And then I teach a course at Roma, and in La Sapienza Rome, and that is a course for masters in neurobiology. And that is even more terrible, because they can understand the molecular pathways, the synaptic transmission, the BDNF, all the pharmacology. But when I say, okay, let's try to do a model, they are scared to touch their new computer. And so it depends on your background. I don't know if this is the answer, and I hope you are not a biologist.
I am.
But this course could give you a good starting point.
But that's the point. I mean, if you want to do science nowadays, you need to be able to use a computer.
Yeah.
At least in a basic terms.
Yeah, absolutely.
That's what those biologists are afraid of. Don't be afraid.
So one of my first students was a psychologist. So he didn't know anything, but he was extremely motivated. So it took a few years, but we published a lot of papers. Now he's a program officer at NIH. So his entire career, it took several years. But if you are motivated, there are no boundaries. Okay?
Any other question?
Maybe later at lunchtime. They are afraid to make questions in front of everybody. This is usually what happens.
Well, I think one important thing to note is that Michele added some perspective to modeling. Because when you start with the idea of modeling a single cell, you say, well, it's nice, I can make a nice model, it fires in the proper way, and looks like the real cell, which is good. But at the end of the story, it seems like something that it is worth, well,
A publication.
Yeah, a publication. You publish a paper, you're happy, but actually the idea is to model the whole story and to get something useful out of this model. So the idea to have the other model is just beautiful, but you can also get something out of it. You advance science and you can really have something useful out of it. So that's why I wanted to thank you.
Yeah, good. Thank you. Thank you for inviting me to see you.
Well,
it's
thank you
nice
for
to
being here.
interact with students every time. So it's, okay, good.
Okay. So one small announcement is more of trouble. So first of all, I apologize.